the title that I was given was asking the right questions and finding and realizing your research interests. So I will say that looking back through my background, I kind of want to give you a history of kind of how I got to where I am, and then what it means to juggle these jobs that I have. And so I think that that's the bigger part. And so along the way, I'm going to have things that kind of just intersect and are kind of be almost non sequiturs, but that's kind of what my life is like. And if you're ultimately going to be a neurosurgeon, you're ultimately going to be a researcher, you're ultimately going to be a mentor. That's exactly how all this goes. You're constantly juggling these balls, so to speak. Please feel free to interrupt me at any time. I'm a pretty casual person once you get to know me. Since I didn't have a talk like this put together, I was trying to think of what my talk would be called. And so I came up with this title, Kicking Ass in Neurosurgery, Research, Mentoring, and Life. So hopefully that doesn't offend anybody here, but I'm pretty casual. I consider myself a blue collar neurosurgeon. So again, please feel free to interrupt me along the way here. I don't think I have any disclosures for this. This is just some formal stuff that sometimes I do talk about some of this lab stuff, but I don't think I will today. So I don't think that that's going to come into play. So, one of the most important things is family. I always keep that in mind. Obviously neurosurgery is a job, research is a job, taking care of patients, all these things, but your family is the most important thing you have. So I always keep that in the back of your mind. And I can tell you that I wouldn't be here today without my family and support from my family. And having a family that supports me to do what I need to do is really important. So my wife, Tina, is in the center there. I can say it hasn't always been easy. My wife does not like my job. Flat out. There's no spouse that's going to say, I want my husband to be a neurosurgeon that's busy 24/7. But my wife understands my job and understands, what we do and feels it's very important and supports me in this. Okay. And then obviously my kids, it's actually funny. Sean, this was a couple of years ago now. Sean, my middle boy, is a couple inches taller than my wife now. So, and my daughter's heading off to college this year. So actually my wife and her are campus shopping this weekend, gallivanting across the country. I didn't realize this. I mean, I realized it, but we're going to be empty nesters at some point in time. It's amazing how, when you start having a family, you don't realize that you're kind of stuck in the quagmire. And I can say that neurosurgery residency and this process is the same way. It's a long process, but at the end of the day, you are going to finish up. You are going to get your job. You're going to realize, your dreams and potential. So, for those, I always ask people when I meet them, I'm like, what's your background? I have a CV. That's not what I'm looking for. Where are you from? What kind of makes you tick? So I'm Minnesotan. I was born and raised in Minnesota. And I can say being at Mayo Clinic is easy for me to do because I'm from here. It's not for everybody. At the end of the day, I'm an adrenaline junkie. I love high-end things that are risky, so to speak. I like climbing to the top of the mountain and skiing down it. I do heli skiing, backcountry skiing. I've always been involved with sports. If you look at our bios, the residents probably put this on my bio, but I have won a bodybuilding contest and have been, performed at the high end of several different sports. Endorphins and adrenaline is kind of the things that make me tick. And I can say that that's sort of a constant theme that you'll see with neurosurgery. So this is a little bit of background that we just briefly covered. I have a 50% research appointment at Mayo Clinic. And I can say that's the most important thing about my job is the time that my department gives me. And in fact, it's actually not 50% on paper. At the end of the day, I actually list 65% research. That's because the NIH requires a certain effort, a commitment to the grants that I carry. Being program director requires 20% commitment to ACGME. So you do the math. I'm listed as like, 15, 20%, surgeon. But Mayo gives me that time to invest into these other opportunities. And so I think I really have to be thankful for having the type of job that I have in an institution that supports me to do that. I did a fellowship at St. Jude Children's Hospital in pediatrics, and it was really to just do pediatric brain tumors is what I specialized in. And I got to spend some time in the lab there and also operating on kids with brain tumors. And so they really helped kind of fine tune my neurosurgical skill set. Not so much my research end of things, but I got to see how St. Jude worked and a little bit of about the research that was going on there, which is top notch. And that has been helpful. Again, residency at Mayo Clinic. and I have a PhD in medicinal chemistry. And that I'm going to bring into play as we kind of go along, as we talk about some research and understanding that you need to play to your strengths at what you're doing. So I have 3 jobs, and that's the reality of it. Job 1 is to be a pediatric neurosurgeon. That's what I was hired to be. And you have to start to focus your practice if you're doing so many things. And so I really am sort of a one-trick pony now. I can do everything. I choose not to do everything because I need time to do other things. And so I really just do pediatric brain tumors. And then when I'm on call, things come in. So I guess 2 nights ago at 10 p.m., you're doing a shunt. That's just part of the business, so to speak. I'm also associate medical director for pediatric trauma for our hospital, and I'll kind of run some of the trauma aspects of that. I guess from an organized neurosurgery part, I also for the pediatric section, I organize the research component of the pediatric section. And so the NREF grants, if you're putting an NREF grant, I help review those NREF grants. That actually goes through these sections. And so there's lots of kind of odds and ends that go on with that. On the research side, I run a basic and translational lab. And this is the part that people don't tell you ahead of time. If you want to be a PI of a lab, if you want to be the person running the lab, it's a small business. You are a small business owner. You're a CEO. And I think that you sort of know that, but you don't know that. What it means is, guess what? You got to deal with the budget. You got to hire people. You got to get money. And I can say that those are the things that nobody's going to teach you. And at the end of the day, I'm constantly screwing it all up. I'm looking at my budget and I'm like, oh, I overspent $10,000 on this grant. And then I got to pay that off somehow, but I don't have any other grants that can cover that grant. And so it's this constant juggling. I have an administrator that I pay. I have a lab manager that I pay. And I think I'm almost to the point where I need to have an MBA person, a business owner almost help me run the lab, because it's just kind of gotten to that size that it would benefit from that. So we're going to get into some of the aspects of research as we go along here. And from education, I'm program director now. I've been doing this now for about 1 year. I've been the associate program director for quite a while, and I've been involved with our education team, both as a resident and a staff. So this is my 8th year as being staff. So stepping into the program director role wasn't new. It was just like a continuation of what I was doing. I also help run our MD Ph.D. program. I also help run part of the aspects of graduate school. And so it's at many different levels, from college students, prep students, medical students, residents. Those are the people that I'm mentoring. And it's a never-ending parade. I think there's constantly somebody always at my door, always at my email, can we talk? Can we go over this? And I can say yes. We'll find the time. It might be at 8 p.m. at night. It might be at weird hours. It might be on the phone, might be on Zoom, might be in person, whatever. We'll find the time. But that's part of this educational rubric, that we have. So the day job, I'm just going to briefly kind of cover this. You guys want to go into neurosurgery, and this is, neurosurgery, driven, symposium. And so that's what I just consider my day job. That's what I was hired for. And so that is sort of a never-ending parade. As a surgeon, you have commitment to your patients that's unlike anything else. Other doctors can go home. You're a radiologist, you read films, you go home, you wash your hands from that. Guess what? You operate on a patient, they're your patient. Those families want to see you. The patient wants to see you. So they get in trouble. They get a complication, something new's going on. You're the person that they want to see. So at 7 p.m. at night, you want to be having dinner with your family. It's a little bit of a different story as a neurosurgeon. So I'm not going to spend too much time on this, but people ask me, what does it take, to be a neurosurgeon? And I can summarize it in one word. It's called sisu. I'm Finnish. My parents were born in Finland. And I grew up with the Finnish culture. And that's not surprising. I guess Minnesota has a really high Scandinavian population. But this word sisu in Finnish, if you try to translate it into English, it comes out as ultimate determination, fortitude, and persistent, carried to an unfathomable level. Okay, so it's the intensity, it's the excellence, and it really doesn't have a comparator word in English. But it was a word that I grew up learning and hearing about from my parents. So I do backcountry adventuring, and this is actually, my dad is the gentleman out here in front, a couple of friends here, and we do 65, 70-mile loops in Canada. and you come across things like this, like these, you're supposed to be on a river and the river dried up and you got, I mean, it's just like, it could be miles of just, just trying to figure out how I'm going to get through this wilderness. My dad slipped and fell, and he's still upset with me because I took this picture. That was my first reaction, was to laugh and take a picture of him on the ground as opposed to go help him up. He's in his 70s now. So I think that was, that was kind of it. But so, having be adventurous and having this drive and determination that's carried, to this end level, that's what it requires to be a neurosurgeon. And I can say that that's the field that people are signing up for. I can sit down with somebody, we get a lot of people applying to our MD PhD program, and I constantly hear, I want to go into neurosurgery. And that's one of their interests of coming to the MD PhD program, Mayo Clinic, is that our MD PhD program is run by a lot of neurosurgeons, and they see that. And I can tell you within, 2 minutes of talking to somebody, yes, no, you can ferret it out. You can see people's kind of intensity and commitment to excellence that they have. And it's going to be like, this person gets it, this person, no, it's probably not going to do it. So again, it's this drive that's inside of you, and that's why you're here. So as I was kind of mentioning, if you want to be a surgeon scientist, you need to focus what you're doing. And, so I can do fetal surgery, myelomeningocele fetal surgery. I can do, instrumented spine surgery. I can do a lot of things in the operating room, but those things will take time. And so be able to have that extra time to spend to the lab and to mentoring, I had to cut out the rest of that stuff. And so there was a point in time where I was the only neurosurgeon at Mayo Clinic. We'd lost one of our other neurosurgeons, and now we have 3 of us. But to be the only pediatric neurosurgeon in an institution is daunting. And so it's very busy. And I was surprised I was able to keep my lab going, but it wasn't healthy, and I couldn't grow my lab properly. So it was being able to get partners in to be able to help out, with that. And so I had to figure out ways, what am I going to do from a research standpoint that I can also integrate with in the operating room? And that became tumors. And I was interested in tumors. I was interested in the molecular pathology of tumors. People ask me how I got interested in neurosurgery. That was during my PhD time, inserting catheters into mice brain. So I developed opioid narcotics. We were trying to develop narcotics that didn't have tolerance or dependence. We did that in rodents. We were able to get rid of the tolerance, the dependence, and the drug addiction from standard opiates. It didn't carry through in high primates, but we were able to actually successfully do it in small animals. But it was doing research on animals, putting catheters into the brain, delivering drugs to the brain, that I was like, well, this is, if I want to do this in patients, I need to be a neurosurgeon. And so that's what kind of brought me into the field of neurosurgery. And so that's why I got interested in neurosurgery. People ask me about pediatrics. How did I get interested in pediatrics? That's a long story, but I can tell you I had a lightning bolt moment for sure. I'd met Rick Boop. Rick Boop was the gentleman who's now stepped down at St. Jude Children's Hospital, but he came as a visiting professor when I was like a PGY3. He gave, a great talk on, pediatric brain tumors, specifically ependymoma, and he showed me these, big, tumors in these small kids. And I was like, wow, that's pretty impressive. But it was when I was studying for my written boards. Actually, the primary boards were just yesterday, so congratulations to any of the residents who went and took that. I was studying for my written boards, and it's a stressful time. Mayo Clinic really wanted us to get really high marks, like top scores. And so I was like, okay, I'm going to really grind it out, studied weeks on end to do this. The day before the exam, you're trying to broadly cover all these topics, and you're just trying to touch on them just for your final review. And I was at a coffee shop, and I came across pediatric brain tumors, and ATRT was like on my bullet list, and it said loss of INI1. And I'm like, cool. And then I'm like, well, what does that mean again? And I'm going to tell you, 45 minutes, an hour went by that I sat there and Googled ATRT and molecular drivers and epigenetics and understanding this chromatin BAF remodeling complex. And I swear to God, I got like a thunderbolt moment where lightning hit me, and I got like a jolt across my whole body, and it said, hey, dumbass, you love this stuff. This is what you really enjoy. Pediatric neurosurgery is it. It was that moment that I was like, okay, I mean, I really like the tumors. I like the molecular biology of the tumors. I like taking care of these patients. And so for me, it was, that this, eye-opening moment that this was it. Not everyone's going to have that, but for me, it was a clairvoyant moment that was just there. So that's a little bit of a tangent, but that's sort of how I kind of got involved with this. You're going to see the research that I do comes back around to the stuff that I already had been doing, and trying to find ways to play to your strengths. Okay, so everybody's going to have strengths. You need to play to your strengths. So when you see the type of research that I do, I'm going to look back and say, well, geez, I was doing something like that during my PhD time. So again, this theme of being excellent, you always, want to strive for excellence. And that's, when people ask me, what am I looking for in a resident applicant? That's it. I'm looking for somebody who wants to be excellent and take it to the next level. Okay, so I'm going to kind of breeze through this part here. Brain tumors are now the leading cause of cancer death in children. That's just a nice fact to know. I mean, kids dying of cancer, they're dying of brain cancer. Okay, so we need to do better. Medulloblastomas, ependymal tumors, high-grade gliomas. I personally do about 70 to 100 pediatric brain tumor cases a year. And then I probably do another 25 miscellaneous cases. So I do 125 cases a year. That number's puny. Most neurosurgeons do a lot more than that. My departmental chair asked me my review, it's like, do you know what your RVUs are per year? Do you know how much this or that? I'm like, nope. Do you know how many RVUs a pediatric neurosurgeon? Nope. So I don't know any of that stuff, because that's not, I operate on people that need it. Okay. You got a brain tumor that needs to come out, and I'm going to take care of you. And so I can say that that is helpful for me to be in a system like that. I'm not working for money. Yeah, I have to, pay the bills, and Mayo Clinic needs me to do cases and stuff. But again, it comes down to having an institution that's going to support me to do that. Okay. So this is the adrenaline of neurosurgery. And this is the stuff that it does keep you coming back. And it's sort of the, I like doing these cases. I mean, this is like a kid that's under 1 year of age. This is a choroid plexus carcinoma, occurs in the ventricle, but you can see the size of this tumor. I call this the jack in the box. You have to know what's going to happen here. When you do your craniotomy and you open the dura, what do you think the next thing that happens is? Okay. So this kid's under a tremendous amount of pressure, and you're going to have a brain that is coming out right at you. And that's exactly what this did. And you got to be, this child has small blood volume. You got to get in and around this quickly. And you got to have a surgical team that can handle this. You have to have anesthesia that's got blood in the room. You have to know how to do these things. So this is just crazy, because you open the jack and you open the box, just comes out at you. You got to get it out quickly. And then the box is deflated. And then you got this, and then you're worried about all these, this is a transverse sinus here. You got all these bridging veins, going into there. So there's lots of peril there. But at the end of the day, you walk out of some of these cases, and, you got your adrenaline fix, for the week after doing something like that. You need to find something to be excellent at. And again, within the field of brain tumors, I decided that I really liked third ventricular tumors, pineal region tumors, suprasellar tumors. And so I've just kind of helped do a lot of these cases, but try to pioneer some of that surgical technique. Keep doing it, and then you kind of get known to be that person that gets to do that. So then you start getting referrals across the country. You write papers on this, you get, people then, seeing what you're doing and stuff like that. And so that's sort of the helpful part. So this is stuff that I really like to do. This is a 17-year-old with the pineal tumor. I can see this on a cell from across the room. This is a mixed germ cell tumor. I just know that. I see the pattern so many times. This will start, you start treating with chemo, then the problem is that these don't always melt away, and then you got to go in there and surgically take them out. And so I enjoy these type of cases. So you need to build part of your practice to say, what do you enjoy? What do you like to do? And then kind of build on that. So I've published probably the most amount of papers on that topic of germ cell tumors over the last handful of years. I teach a class with dissection for our anatomy residents that we're now going to be kind of offering to residents across the country, hopefully soon. And then, then people start to notice that, and then you get on neurosurgical focus. We're doing pineal tumors. Can you help us be one of the editors for that? And so then you just kind of become, one of the leaders kind of in that field. So you got to kind of, find out what you're interested in, and then you got to, build on that. You got to publish on that, and then do other things that are going to set you apart. And so I really kind of enjoy that aspect. I don't want to spend too much time on that, but again, it's sort of finding a niche kind of within the niche, that you enjoy. How do students get involved? And I can say that you need to find mentors. And so, people, find me as a staff, but guess what? Student mentors are some of the most helpful people that I had, going along the way as well. So if you're a medical student, finding a resident, PGY 1, 2, 3, somebody that can tell you how they went through the mix. And, college students, finding medical students, it's, just goes along the way. You need to have that right mix of mentors that are there. And it's going to be people that have time just to sit down and talk with you, pointing directions, and can, answer questions. That can be in person, over Zoom now, electronically over email. And so but the onus is on you as the student learner to find those people. But don't be shy. I can just tell you that if somebody's going to blow you off, then they're not going to be a good mentor. I will say that, I drop the ball routinely on trying to get, some of these meetings set up. And it's because I get so busy that I'm just like, oh yeah, remind me at this point in time. And I always tell people, please, if you didn't hear back from me, just please email me again. And it's just because, so many things get busy. But I think it's just reaching out to people, and again, but don't limit yourself just to look to a staff. It's, everybody that has kind of gone through some of the stuff that you're looking to go through are the helpful people there. The reason why I put this in there is because this is what neurosurgery life is. This is me at Banff with my family. I don't know, it's probably 4 years ago. And as I'm taking this picture, my phone is ringing, and it's the ER calling me. Okay. So, when I look at this picture, I just remember being frustrated, dealing with the patient in the emergency room while I'm trying to take, picture of my family at Lake Louise. I'm never going to probably be back here with Lake Louise with my family and stuff. So, but, I got to say time out. Okay. And honestly, this doesn't really bother me, but then I got my wife who's just like, well, just typical stuff that you're always dealing with and stuff. So, but it's just, that's why I put this here, is that's what being a neurosurgeon is like. You buy these patients, you own these patients for the rest of your life. Okay. And again, pediatrics is in such a way that hopefully they have a long life. And so it's, it's just this commitment as a surgeon that, you're their doctor, and they're going to be able to find you and stuff. So I think that's the commitment level, that's required. So let's transition into research. So again, you have to overlap what you are doing in the operating room to some degree with what you're doing in the lab. And so for me, pretty much any malignant tumor, rebiobank. So retake tumor specimen and refreeze it, retake tumor specimen, try to grow it in a petri dish, retake blood, retake urine, retake CSF, and try to biobank this. Ultimately, what I'd like to do then is, figure out in the lab the way these things tick and find new therapy that we can bring back to the patient. So what I'm describing to you is translational research that, comes from the patient, that goes to the bench side, that hopefully goes back to the patient in some fashion. And so that's where, as a surgeon, I'm collecting these samples that only a surgeon can. And also some of the things that we do from the translational parts are only things that surgeons can do. And I'm just going to quickly, speak on that. I'm not going to bore people with the research aspect. I don't know how many patient-derived cell lines we've made, it's a lot now. This number just keeps growing. We probably have 5 or 6 now, DIPG cell lines. So what do I study in the lab? I study diffuse intrinsic pontine glioma. Now it goes under the name, diffuse midline glioma, histone altered, but it's this H3K27M tumor. We also study ATRT and a couple other things, but by and large, it's mostly this DIPG. And I didn't plan on getting into DIPG research, but I just want to briefly tell you how did that happen? So I didn't pick this research. This research picked me. And I think it's also the way that I picked pediatrics. It's sort of, my interest just became that's just what it is. But again, I did not choose DIPG research. And in fact, I was gearing up to do other stuff, but as a first year staff, I saw a kid, his name's Jacob, put the little eye patch over there for a reminder. It was the first DIPG patient I took care of. He showed up at my clinic with an eye patch. He got double vision. And he had a pontine tumor and rebiopsied it. And it had atypical features, so it just wasn't a slam dunk DIPG tumor. It came back as a diffuse, glioma. We didn't have this H3K27M histone altered tumor at that time. So he was just labeled, diffuse, brainstem glioma is what his label was. But I knew it fell under the rubric of DIPG tumor at that time. And I remember seeing him back in clinic and going over the diagnosis with him and his family. And I remember Jacob, I'm just telling him, well, this is a diffuse, glioma in the brainstem, and we need to move on to radiation now. But Jacob, looks at me and he goes, am I going to be all right? And I can just tell you to this day, I still get choked up thinking about that, because the answer is no, you're not going to be all right. You're going to be dead in 1 year. Every one of them is dead. And so this is a disease that we can't treat. We have no treatment for this disease. Anyway, a part of our biobanking efforts, his sample came to my OR and it grew as my first cell line. And it had the histone mutation in it. Retrospectively looking at it, we knew that mutation was out there. It just wasn't used from a pathology standpoint yet. And so that's when seeing the clinical story, watching him die, and then also having a surgical specimen from a pre-treated patient. So most of the DIPG samples that people have out there come from autopsies, because people haven't been biopsying these up front. We are now more now, but we weren't 8 years ago. And so having this sample and watching this kid go through this, and his family go through this, that's, again, I didn't choose this. It chose me. Okay. That's how I got involved with it. Our lab has developed a lot of these cell lines now from patients, and we've developed new types of xenografts. So we did zebrafish at one point in time. That I'm going to say that's not too, it's hard to do studies in zebrafish, because how do you dose drugs? You're just going to put them in the water, but we did do that. We've been working over the last 4 or 5 years with this chick embryo model. It's pretty cool. You crack a fertilized chicken egg, and you can put the tumor on there, and you can do drug studies with that. So we've published kind of our routes onto how to redo our xenografts, both in rats, mice, these chick embryos and stuff. So our lab probably has some of the most robust experience developing, these models that we can use, for testing. This is my lab here. A couple of people are missing. This was actually just a couple of weeks ago. I try to do a lot of fun things with the group. So, every month or so, I want to get together, we do a lunch, go dinner, and happy hour, try to have people over to my house. Just briefly, Cody Nesvik is a resident. He's a PGY6. He spent 2 years in the lab. He had an NREF grant. Animal tech postdoc, MD, Ph.D. student, college student, me, graduate student. That's my lab manager, graduate student. And then I have a postbac and another postdoc. So that's what kind of fills out the lab now. I want everybody in my lab to get along. I want the lab to be kind of a light atmosphere where people don't feel like it's a sweatshop. Do people work hard in my lab? Yes, but I want it to still have a kind of a light atmosphere. Again, these were just the people that I just kind of rattled off. This is my funding. I have 4 NIH grants, 3 of them, this R01, R61, K08, I'm the PI for. So I can say that it's going to be rare for you to find a neurosurgeon that has their own NIH funding like that to that degree. And I can say that most neurosurgeons are going to collaborate with people and be co-PI, be co-I, and that's how they do it. But I really enjoy being the principal kind of investigator. I enjoy being the person who drives the lab and the research. And so that sort of takes it to a new level. And that goes back to you have to find your own, this is a small business. You are, finding your own research money. You're hiring the people, you're firing the people, you're managing the budget. And so that kind of goes back to that. These are projects in the lab. I'm going to touch on kind of one project that kind of overlaps with stuff as being a neurosurgeon and plays to my interest. And so this is what DIPG tumor looks like. It's the, expanded pons. It's an infiltrated tumor. There's been 100+ clinical trials with drugs. Every one of them has failed. every one of them has failed. Every one of these kids dies. Again, it's a death sentence. It was 2012 that histone mutation became known. And it was found that this histone mutation is found in these midline portions. So it turns out it's not just the pons, but it's the pons, the thalamus, and the spine. The pons is the most common spot, but this tumor lives in the midline structures of the brain. There's other mutations. There's another mutation that occurs in the hemisphere, which I'm not going to cover. What my lab really studies is just this midline H3K27N mutation. It has this mutation. We can pick it up by immunohistochemistry now, but it also has this loss of K27 trimethylation. I won't bore you with the details of that. So these are some of the things that you're asking, questions, and these are things that my lab looks at. How mutations in the genome reprogram or drive the epigenome, to drive tumorigenesis. So Cody spent, 2 years doing ChIP-seq and a whole bunch of, what I would call basic science type of stuff. Diagnosis: can we do a liquid biopsy? Can we find this in the CSF and in the serum and stuff? And the answer is yes, we can, but that's a complex. Therapeutic vulnerabilities: we spent a lot of time doing drug screens, but in the end of the day, we need to translate our efforts back to the patient. And so that's what I'm going to show you here, is taking some of the stuff that we've done from the above portions of this and trying to get back into the patient. So you have to ask yourself, why has every clinical trial failed for DIPG? Is it the wrong drugs? I'm going to tell you that's not the case. We have drugs for cancer and they work. Okay. But is it we're not getting the drugs there? And so that's where direct drug delivery, so convection enhanced delivery, there's a MRI guided focused ultrasound, there's other techniques that are kind of coming up that people are doing. But I'm going to tell you that it's actually more complex than that. And this comes from some of the stuff that we've done in the lab. Oh yeah, so again, non sequiturs. So you're just minding your own business and you're like, what's this? This is every parent's worst nightmare too. You tell your kid not to run around with a screwdriver. That's what that is. So you're minding your own business on a Saturday night and you get called in to come take care of this. So again, just as a neurosurgeon, these are things that you're just going to be dealing with in the middle of everything else. It's like, there it is. So, okay, again, non sequiturs. Convection enhanced delivery is a method to deliver drugs directly to the brainstem. Okay, so it's a way that we can bypass the blood-brain barrier and get drugs directly there. This has been, so a phase 1 study just was completed. Mark Swedane led that, that study and showed that it was safe to do. It didn't look at efficacy, but now we can say that, CED is safe to do in a large, cohort of kids, but we can directly deliver drugs there. People have looked at catheter design and how to do this. But I can tell you, people haven't looked at what are the drug properties that are necessary to be giving the drugs directly. So how does molecular weight, lipophilicity, efflux pump, drug clearance, how do these things play into to convection enhanced delivery and just even drugs in general, to the brainstem area? And so we spent a lot of time looking at direct drug delivery. And so this is a little bit of what some of this looks like. This is rat surgery, where we did convection enhanced delivery. We also did diffusion-based delivery of increasing molecular weight size drugs, essentially. The bigger stuff was just Fitzy dextran beads, but then had smaller stuff. But we looked at how these drugs distribute based on their size. And this was one of the first papers that we published with CED. So the point is, now I'm going to talk about finding the right drug. So our lab has done a lot of drug screening, and so a lot of it's just done with cell viability. You just screen FDA drugs, epigenetic drugs. You have these big libraries, and you try to find drugs that work. CRISPR, shRNA screening. That's where you knock out signaling pathways and say, are these signaling pathways important in these tumors and stuff? So we've done all of this stuff, and we've had some of our key hits. And I don't really want to discuss our key hits other than I'm going to pull out an example drug here. So this is Erica. She's a 4th year graduate student now, and this is essentially her Ph.D. thesis. And this is one of her big manuscripts that is going in now. I guess all this is unpublished right now, but this was an epigenetic drug screen. So basically drugs that we know modify the epigenome or interact with chromatin and such. These were all Aurora kinase inhibitors, and they were all potent drugs. So all of the Aurora kinase inhibitors came out as potent hits in our DIPG cell lines. alisertib is already been in all these, the alisertib, barisertib, and this AT drug were all in clinical trial. One's an Aurora kinase A inhibitor, one's a B inhibitor, one's a PAM inhibitor. So we started focusing on these because they're already in clinical testing, so to speak. And we did a bunch of studies that showed that this Aurora kinase certainly is important in these DIPG tumors. That's what my R01 actually focuses on, is that importance of Aurora kinase. So I can tell you, we feel like we found a targeted therapy. The Aurora kinase is phosphorylate serine 28, which is right next to the lysine 27. And we think that there's this epigenetic interplay there. Again, don't want to cover that. But what I want to tell you is that we took alisertib, we treated a DIPG flank tumor with it, and we decreased the tumor size. We show on-target effects. So we're happy, great. We can decrease the tumor in vivo. Well, that's a flank tumor. We go into Susie Baker from St. Jude has a GEM model, which is a spontaneous H3K27 forming tumor model. And we give alisertib orally, and we don't see any difference when we give this orally. Well, guess what? The alisertib is 3% brain penetrant. Okay, so when we actually test how much blood or how much compound we get in the brain, is really low. So then we go, let's give it by CED. So give it directly to the brain, and we gave CED several times per week, and guess what? We still didn't see a benefit. Okay, so then you have to go back. So again, I have a medicinal chemistry background, pharmacology. So then it's going to be like, what's happening to this drug? So we start looking at the clearance rate of the drug, and this panel D here is at time 0. If when we directly inject the drug, we get to this level of drug. Within 1 hour, over half of the drug is gone. The half-life of the drug is much longer. The drug's been cleared. The drug gets out of there super quickly. We found this with other drugs. Panabinestat's actually in clinical trial for CED. People are giving it by CED. It clears within 2.5 hours. We checked a couple of other small molecules, same thing. 1 to 2 hours seems to be the clearance rate for these drugs. So these drugs are getting cleared super rapidly from the brain. So we started looking at, can we block these efflux pumps, because they're sometimes responsible for this? Can we infuse drugs slowly over a period of time? Can we develop nanoparticle formulation? Can we chemically modify these drugs to get there longer? So here's a long, slow infusion over a week. We can cure a subset of those animals. So, because again, we found what we think is the right drug, but we weren't getting the right drug there long enough to have the appropriate effect. Okay. And so that was our take-home message. And I think that's what it's going to take to actually make a difference in this tumor. So one of the things we're doing is nanoparticles. So this is nanoparticle delivery, where we're encapsulating nanoparticles, and we're getting about maybe 24 to 72 hours of drug available with a nanoparticle CED delivery. Another thing we're doing is we're pegylating drugs. Pegylation is a way that you can actually keep a drug there longer. We came up with this interesting concept of coupling the drug to dextran beads, and that actually makes the drug stick around longer. So we've done this boring dextran. So what you see here is chemistry. What you also see here is a surgeon implanting a catheter into the brain. I'm playing to the strengths that I have and that I even had during my PhD time. And I'm bringing those things into what I'm doing now. And again, I know people won't quite have the background and stuff, but you have to play to what you're interested in and what your strengths are. And so that's what I was trying to bring to the field. What do I have to bring to this field to be able to do this? I need to fly through this. So I'm just going to also show you how life creeps up. This is me. I have a broken shoulder. This was probably 5 years ago. I fell off a bike. My kids were chasing me. This is me last weekend. I have an avulsion fracture. I was playing ice hockey with my boys. I have an avulsion fracture in my 5th metatarsal. So this was actually me this week in a booted cast. I can't stop working out though, because of course that's what makes my brain run appropriately. I know I only have 2 more minutes. I wish I'd left. I probably should have skipped through this, the mentoring aspects of things. So I manage residents. We have 25 residents in our program. I help manage the MD, PhD folks as well. I spend a tremendous amount of time mentoring people. This is the advice that I give to these people. Again, be excellent at what you do. Strive for excellence at what you are doing. That really is it. If you want to be excellent at something, that's what it's going to take. And so, you have to find what your passion is, but be excellent. Publications, you need publications. I hate to say this, there are publications for those that can count. There's publications for those that can read. Okay, I look for both, but most program directors just have a number. They're going to triage your applications based on number of publications. Okay, so you need publications. And sometimes counting is more important at the level that you're at. The level I'm at, counting doesn't matter at all. I'm looking more for publications. I have plenty of publications for those that can count. And I still keep getting those publications. Why? Because you folks need those publications. So I help, with those. But again, it's sort of a vicious cycle to some degree, but you need them. You need to have publications. Your letters of recommendation, don't look for big names. Okay. You need somebody who's going to write you a genuine letter of recommendation. When I pull up this and I see person X biggest name and blah, 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 and it's the same recommendation that he's given for 10 other people, that's not helpful. What's helpful is somebody that I may not know that is like, this person is the best thing since sliced bread. That means way more. Get to know somebody, get to know a neurosurgeon, get to have somebody write you that genuine letter of recommendation. Here's my favorite advice. It's a marathon, okay? How do you run a marathon? You pace yourself. What if you want to win the marathon? You race it. So you have to pace yourself, yes, but it's still a race. And as neurosurgeons, we're the people sitting in front of the race, okay? So that's an important thing. Work-life balance. I don't believe in work-life balance. Work gets in the way too much. And what I call it is work-life integration. I take my family with work trips. We went to Hawaii in January. My family came with me. I was at a meeting. Okay. Downtime. My kids play sports. I'm sitting there waiting an hour. I drop them off. I got to wait for the game. My computer's in my lap. I'm working. Driving. My wife hates this, but we're driving to like a kid's like whatever, or we're going to the airport and I'm on the phone getting everything done, because that's what it takes. You need to protect important days. And if that means taking some days off, that's what it means. And I've had to do that. Like if I don't want to miss a kid's birthday, I literally have to say, okay, I need to take this day off. I don't do that now. Like kids' birthdays aren't it doesn't seem to be as critical, but when they were younger, I actually did take those days off. So I just knew that I would just have, and I still probably work during those days, but I just needed to have an out where I just like, okay, I'm going to have some time here. So take home points. If you plan to be a physician scientist, you got to overlap kind of what you're doing as a surgeon and what you're doing as a scientist. You got to play to your strengths. Translational research is really the key. Okay. I do a fair amount of basic research too, but it's really the translational stuff that's the important part. I want to make a difference. At the end of the day, I want to find something that is going to help these poor kids with DIPG tumors. Pick whatever field you're looking at, but make that difference. And it's in the patient that find the right mentors. That's important. It's finding the people that are going to spend time with you and help you through the process. Your CV is important. Your CV is what we look at. So that's number of publications. That's, grants, awards. That's what you've done. That is important. You need to build a strong CV. Okay. And then again, excellence. And that's it. I just tell, what are the number one thing? I'm looking for people to be excellent. So that's it. Thank you very much.
Hey everyone, Ryan Rad here from neurosurgerytraining.org. If you like that video, subscribe and donate to keep our content available for medical students across the world.